So thanks for the introduction. It is my pleasure to talk with you and be in this panel. So today I will talk a little bit about the impact of the matrix on phage predation of Staphylococcus epidermidis biofilms. So just a brief introduction on the organism. Staph epidermidis is a coagulase negative staphylococci that inhabits in our mucous membrane and skin. However, it has become one of the most predominant causes of nosocomial infections worldwide, namely in indwelling medical devices. These infections are particularly difficult to treat for two main reasons. One, some of the Staph epidermidis strains can be resistant up to 8 different families of antibiotics, and Staph epidermidis is known as to form thick biofilms, which is indeed considered its major virulence factor. Just a brief definition of biofilms. They can be defined as bacterial communities attached to a surface and surrounded by a self-produced exopolymeric matrix. And we are working as phages as potential alternatives to treat bacterial biofilms. However, when phages interact with biofilms, there are several things that can happen that can affect this interaction. So, for example, some strains have prophages, and it is described, for example, for Pseudomonas aeruginosa, that prophages can impact the biofilm formation. Biofilms are composed by dormant cells, which are cells that are with low metabolic state, and it impairs the infection of the majority of the phages. Also, there are some discussions about the presence of resistant cells at the beginning of the biofilm formation, quorum signaling. Also, the biofilm matrix and outer membrane vesicles can act as a decoy for phage predation. On the opposite side, phages can use some of the biofilm structure to migrate on the biofilm, so they can use the channels to reach the bottom layers of the biofilm. And for example, some phages are equipped with depolymerases that, besides degrading the capsule, in some cases they can degrade the EPSs and accessing more easily the biofilm cells. So the story that I will tell you about today began at the beginning of my PhD, which was several years ago. So with the increasing importance of phage therapy, and at that time, and even now, there are not so many phages isolated for S. epidermidis. We wanted to isolate and characterize new Staph epidermidis phages and analyze their interaction with Staph epidermidis cells, namely on biofilms. I will focus this talk on one phage, which is CEP1. At that time, this was in 2013, I had the pleasure to visit Dr. Ackerman's lab, and we performed the transmission electron microscopy of this phage. We noticed that this is a myoviridae phage, and then we tested the lytic spectra. We tested this on 41 S. epidermidis strain, and we could observe that the phage could have an effect in all of the strains tested. In addition, these strains were from different origins and infection sites. No infection was detected in other staphylococci or other gram-positive organisms. The genome was sequenced and annotated, and it shows the structure of a typical Twort virus. However, the homologies at that time were smaller than 60%, and now, after Evelyn's talk from yesterday, I updated this. So this is now a genus of the Helviridae phage, and it's called the Sepuna virus phages from this genus. So to start, we tested the phage on biofilms with the idea that phages that usually phage K or other phages that belong to this type of phages are good on biofilms. So we formed the biofilms on microtiter plates, use the phage. In this case, we used an MOA of 1. And unfortunately, no significant reductions were observed on the cell counts nor on the total biomass. So to start, this phage was not a good candidate to treat biofilms. But as it is similar to other phages, and we wanted to understand the reasons that led to this inefficacy. So we wanted to start to see if this was a problem of the different cell populations that inhabit on the biofilm, or if it was a consequence of the biofilm matrix. So starting on the cell populations, we tested the phage on different planktonic cells. So on exponential phase cells, we used the same MOI. So after 2 hours of infection, we already see some reduction that was even higher at 4 and kept stable at 8 hours of infection. Then we tested the phage on stationary phase cells. And surprisingly, the phage was active against stationary phase cells, although it takes longer to act than in exponential phase cells. The phage was very effective on controlling stationary phase cells, which is a rare effect, and we wanted to understand what was happening. So we performed a kind of an adaptation to the live dye staining, but on a flow cytometer. And this is the population before we had in the phage. On the x-axis we have the intensity of cyber, and on the y-axis we have the propidium iodide. And already after 5 minutes of infection, we already see something changing on the population. There is a change on the majority of the population to an increase of the mean fluorescence intensity of the cyber. And this increase on the mean fluorescence intensity kept growing until the 45 minutes of infection. At 60 minutes of infection, the infection was kind of stable. And after 150 minutes of infection, 3 things are possible to observe. So here we have a huge increase on the cell counts that are positive for propidium iodide, so cells that are probably going to die. We see here an increase on non-fluorescent counts, which are cell debris. And you don't see this in here, but you have to trust me, we see already here less cell counts. So at this point, we know that we already knew that the phage was effective against stationary phase cells very efficiently, but now we know that this happens very quickly. So we wanted to understand what's happening from a transcriptomics point of view. This is not RNA-seq, this is just qPCR. And we used a host gene and 4 phage genes. So at exponential phase cells, we can see the modularity described for phages. So early genes being expressed in the first minutes of infection, nothing happened on the middle and late genes, and they were increasing during the course of infection. Regarding the host RNA polymerase on exponential phase cells, no significant changes were observed during the track of infection. In opposition, on stationary phase cells, after 5 minutes of infection, we already see an increase, an overexpression of the host RNA polymerase, which was kept, which even was more even expressed in the other points of infection. And regarding the phage genes, the modularity was not so seen during the infection of stationary phase cells. So we know, at this point, we know by the part of the RNA polymerase that the phage addition to stationary phase cells somehow activates the cells in order to express the phage genes, which, not surprisingly, does not happen on exponential phage cells. Then we wanted to see if other biofilm populations could also be impairing the phage efficacy on biofilms. So we tested the phage on biofilm released cells. These cells are the cells that are on the biofilm and are released to the planktonic part, and they were described as having a higher virulence potential than planktonic phenotypes. So we formed the biofilms, we washed them 2 times, we had fresh media, incubated for 3 hours, and then with the cells that are on the platonic phase, we infect them with the phage. And we showed that the phage can control very efficiently biofilm released cells. To finish all the types of cells that we tested with this phage, we tested the phage on persister cells that are, as the majority of you know, phenotypic variants that are tolerant to antibiotics, but are described to be responsible for recalcitrance of chronic infections, namely the ones associated with biofilms. So we grow the cells for 12 hours. In this case, it was a protocol, an adapted protocol. We incubated for 48 hours with vancomycin, washed them 2 times, and add the phage. And we observed in this case that the profile was similar to the one we observed on stationary phase cells. So almost no significant reduction during the first 2 hours of infection, but this infection grew at 4 and 8 hours of infection. This is probably due to the duration of the culture. But we also show that the phage is effective in controlling persister cells. So in every planktonic cell that we use with this phage showed to be very efficient on controlling the cells. So at this point, we wanted to understand, as this is not a consequence of the different cell populations within the biofilm, if the inefficacy that we observed that I talked at the beginning of this talk was a consequence of the biofilm matrix. And we did a very simple assay. We formed the biofilm with we applied the debridement with a tip. It was not a full debridement. It was just something to disrupt lightly the biofilm structure. So the matrix was still there, just the biofilm was not fully intact. And then we had the phage. And just with this light disruption, we were able to observe that the phage was active against these scrap biofilms. Of course, a 2.5 log reduction. Of course, it's not a reduction so high as we observed on planktonic cells, but in this case, the matrix is still there. So to understand what's happening in the biofilm, we moved and performed. We developed a probe, a fish probe, using DNA mimics, and we performed it in combination with other fluorochromes and to observe the biofilms on confocal laser scanning microscopy. The probe that we developed was developed to target the Staphylococcus epidermidis cells that are infected with the phage. In this case, the targeted mRNA was the major capsid protein. So we did a triple staining. We used DAPI for staining the cells. We used WGA with FIT-C to target the polyanestyl glucosamine, which is the main component of the biofilm matrix. And we used the probe to show us the phage-infected cells. And this is an example of what we observed. And what we observed in several images that we took is that phage-infected cells seem to locate predominantly in the regions with lower amounts of matrix. And that emphasizes why we see almost no reduction on biofilms, and on scrap biofilms, we always see some reduction. So this kind of, at least for us, proved us that the matrix is impairing the efficiency against biofilms. Just two papers that came out very recently in E. coli, but showed also in other studies using different techniques that the biofilm architecture can trap the phages, preventing viral predation. So to conclude, in this study, we isolated one of the first highly specific and broadest range Staph epidermidis phage. I think we show that the structure of the biofilm difficults the efficacy against biofilms, but in opposition, we show that this phage is a particular phage. It's a phage that I studied for several years, but it's active against stationary, dormant cells, persister cells, biofilm released cells. So it could be a good candidate for therapy for treating biofilms if associated with some mechanical or enzymatic methods for biofilm dispersion, or, for example, by genetically engineer the phage. To conclude, I want to acknowledge everybody that was involved in this work from the beginning, my group, funding agencies,